Now we are going to continue with the renal physiology part. So today we are going to learn about inulin clearance can be used to estimate GFR. If a substance is freshly filtered as freshly as water and is not absorbed or secreted by the renal tubules, then the rate at which that substance is excreted in the urine is equal to the filtration rate of the substance by the kidneys. So GFR into PS is equal to US into V. The GFR therefore can be calculated as the clearance of the substance as follows is equal to GFR is equal to US into V by PS into GFR is equal to US into V by PS is equal to CS. A substance that fits this criteria is inulin, a polysaccharide molecule with a molecular weight of about 5200. Inulin, which is not produced in the body, is found in the roots of certain plants and must be administered intravenously to a patient to measure GFR. Renal handling of inulin. In this example, a plasma concentration is 1 mg per ml, urine concentration is 125 mg per ml, and urine flow rate is 1 ml per minute. Therefore, 125 mg per minute of inulin passes into the urine. Then inulin clearance is calculated as the urine excretion rate of inulin divided by the plasma concentration, which yields a value of 125 ml per minute. This 125 ml of plasma flowing through the kidneys must be filtered to deliver the inulin and appears in the urine. Inulin is not the only substance that can be used for determining GFR. Other substances that have been used clinically to estimate GFR include radioactive iothalamate and creatinine. So next topic is creatinine clearance and plasma creatinine concentration can be used to estimate GFR. Creatinine is a byproduct of muscle metabolism and is cleared from the body fluids almost entirely by glomerular filtration. Therefore, the clearance of creatinine can also be used to assess GFR because measurement of creatinine clearance does not require intravenous infusion into the patient. This method is much more widely used than inulin clearance for estimating GFR clinically. However, creatinine clearance is not a perfect marker of GFR because a small amount of it is secreted by the tubules. So, the amount of creatinine excreted slightly exceeds the amount filtered. This is normally a slight error in measuring plasma creatinine that leads to an overestimate of plasma creatinine concentration. And then these two errors tend to cancel each other. Therefore, creatinine clearance provide a reasonable estimate of GFR. If in some cases it may not be practical to collect urine in a patient for measuring creatinine clearance, an approximation of changes in GFR, however, can be obtained by simply measuring plasma creatinine concentration (PCR), which is inversely proportional to GFR. So GFR is equal to CCR is equal to UCR into V by PCR. If GFR suddenly decreases by 50%, the kidneys will transiently filter and excrete only half as much creatinine, causing accumulation of creatinine in the body fluids and raising plasma concentration. Plasma concentration of creatinine will continue to rise until the filtered load of creatinine and creatinine excretion return to normal and the balance between creatinine production and creatinine excretion is re-established. This will occur when plasma creatinine increases to approximately twice normal. Then, if GFR fails to 1/4 normal, plasma creatinine would increase to about 4 times normal, and a decrease of GFR to 1/8 normal would raise plasma creatinine to 8 times normal. Thus, under steady state conditions, the creatinine excretion rate equals the rate of creatinine production despite reductions in GFR. However, this normal rate of creatinine excretion occurs at the expense of elevated plasma creatinine concentration. Then, PAH clearance can be used to estimate renal plasma flow. Theoretically, if a substance is completely cleared from the plasma, the clearance rate of that substance is equal to the total renal plasma flow. In other words, the amount of the substance delivered to the kidneys in the blood, renal plasma flow into P5, would be equal to the amount excreted in the urine. Thus, renal plasma flow could be calculated as RPF is equal to US into E by PS is equal to CS. Because the GFR is only about 20% of the total plasma flow, a substance that is completely cleared from the plasma must be excreted by tubular secretion as well as glomerular filtration. There are no known substances that are completely cleared by the kidneys. One substance, however, PAH, is about 90% cleared from the plasma. Therefore, the clearance of PAH can be used as an approximation of renal plasma flow. To be more accurate, one can correct for the percentage of PAH that is still in the blood when it leaves the kidneys. The percentage of PAH removed from the blood is known as the extraction ratio of PAH and averages about 90% in normal kidneys. In diseased kidneys, this extraction ratio may be reduced because of inability of damaged tubules to secrete pH into the tubular fluid. The calculation of RPF can be demonstrated by the following example. Assume that the plasma concentration of pH is 0.01 mg per ml, urine concentration is 5.85 mg per ml, and urine flow rate is 1 ml per minute. So, pH clearance can be calculated from the rate of urinary pH excretion divided by the plasma pH concentration. This clearance of pH calculates to be 585 ml per minute. If the extraction ratio of pH is 90%, the actual renal plasma flow can be calculated by dividing 585 ml per minute by 0.9, yielding a value of 650 ml per minute. Thus, total renal plasma flow can be calculated as total renal plasma flow is equal to pH clearance by pH extraction ratio. The extraction ratio is calculated as the difference between the renal arterial pH and renal venous pH concentrations divided by the renal arterial pH concentrations. One can calculate the total blood flow through the kidneys from the total renal plasma flow and hematocrit, the percentage of red blood cells in the blood. If the hematocrit is 0.45 and the total renal plasma flow is 650 ml per minute, the total blood flow through the both kidneys is 650 or 1182 ml per minute. Filtration fraction is calculated from GFR divided by renal plasma flow. To calculate the filtration fraction, which is the fraction of plasma that filters through the glomerular membrane, one must first know the renal plasma flow and the GFR, inulin clearance. If renal plasma flow is 650 ml per minute and GFR is 125 ml per minute, the filtration factor is calculated as FF is equal to GFR by RPF is equal to 125 by 650 is equal to 0.19. Comparisons of inulin clearance with clearance of different solutes. The following generalizations can be made by comparing the clearance of substance with the clearance of inulin, a measure of GFR. If the clearance rate of the substance equals that of inulin, the substance is only filtered and not reabsorbed or secreted. If the clearance rate of a substance is less than inulin clearance, the substance must have been reabsorbed by the nephron tubules. If the clearance rate of substance is greater than that of inulin, the substance must be secreted by the nephron tubules. The approximate clearance rates for some of the substances are as follows, like glucose clearance rate 0, sodium 0.9, chloride 1.3 and creatinine 140. Then next topic, determinants of the GFR. The GFR is determined by the sum of the hydrostatic and colloid osmotic forces across the glomerular membrane, which gives the net filtration pressure, and the glomerular capillary filtration coefficient, expressed mathematically, the GFR equals the product of Kf and the net filtration pressure. GFR is equal to Kf into net filtration pressure. The net filtration pressure represents the sum of the hydrostatic and colloid osmotic forces that either favor or oppose filtration across the glomerular capillaries. These forces include hydrostatic pressure inside the glomerular capillaries, glomerular hydrostatic pressure, which promote filtration. Next topic, increased Bowman's capsule hydrostatic pressure decreases GFR. Direct measurements using micropipettes of hydrostatic pressure in Bowman's capsule and at different points in the proximal tubule in experimental animals suggest that a reasonable estimate of Bowman's capsule pressure in humans is about 18 mmHg under normal conditions. Increasing the hydrostatic pressure in Bowman's capsule reduces GFR, whereas decreasing this pressure raises GFR. However, changes in Bowman's capsule pressure normally do not serve as a primary means for regulating GFR. In certain pathological states associated with obstruction in the urinary tract, Bowman's capsule pressure can increase markedly, causing serious reduction of GFR. For example, precipitation of calcium or uric acid may lead to stones that load the urinary tract, often in the ureter, thereby obstructing outflow of the urinary tract and raising Bowman's capsule pressure. The situation reduces GFR and eventually can cause hydronephrosis or distension and dilation of the renal pelvis and calyces, and can damage or even destroy the kidney unless the obstruction is relieved. Increased glomerular capillary colloid osmotic pressure decreases GFR. As blood passes from the afferent arteriole through the glomerular capillaries to the efferent arteriole, the plasma protein concentration increases about 20%. The reason for this increase is that about 1/5 of the fluid in the capillaries filtered into Bowman's capsule, thereby concentrating the glomerular plasma proteins that are not filtered. Assuming that the normal colloid osmotic pressure of plasma entering the glomerular capillaries is 28 mm per Hg, this value actually usually rises to about 36 mmHg by the time the blood reaches the efferent end of the capillaries. Therefore, the average colloid osmotic pressure of the glomerular capillary plasma proteins is midway between 28 and 36 mmHg or about 32 mmHg. These two factors that influence the glomerular capillary colloid osmotic pressure are the arterial plasma colloid osmotic pressure and the fraction of plasma filtered by the glomerular capillaries. Filtration fraction increasing the arterial plasma colloid osmotic pressure raises the glomerular capillary colloid osmotic pressure, which can decrease the GFR. Next topic, increased glomerular capillary hydrostatic pressure increases GFR. The glomerular capillary hydrostatic pressure has been estimated to about 60 mmHg under normal conditions. Changes in glomerular hydrostatic pressure serve as the primary means for physiological regulation of GFR. Increase in glomerular hydrostatic pressure raises GFR, whereas decrease in glomerular hydrostatic pressure reduces GFR. Glomerular hydrostatic pressure is determined by three variables, each of which is under physiological control: arterial pressure, afferent arteriolar resistance, and efferent arteriolar resistance. Increased arterial pressure tends to raise glomerular hydrostatic pressure and therefore to increase GFR. Therefore, as discussed later, this effect is offered by autoregulatory mechanisms that maintain a relatively constant glomerular pressure as blood pressure fluctuates. Increased resistance of afferent arterioles reduces glomerular hydrostatic pressure and decreases GFR. Conversely, dilation of the afferent arteriole increases both the glomerular hydrostatic pressure and GFR. Constriction to the efferent arteriole increases the resistance to outflow from the glomerular capillaries. This mechanism raises glomerular hydrostatic pressure, and as long as the increase in efferent resistance does not reduce renal blood flow too much, GFR increases slightly. Then physiological control of glomerular filtration and renal blood flow. The determinants of GFR that are most variable and subject to physiological control include the glomerular hydrostatic pressure and the glomerular capillary colloid osmotic pressure. These variables in turn are influenced by the sympathetic nervous system, hormones and autocoids, vasoactive substances that are released in the kidneys and act locally, and other feedback controls that are intrinsic to the kidneys. Then strong sympathetic nerve system activation decreases GFR. Essentially, all the blood vessels of the kidneys, including the afferent and the efferent arterioles, are richly innervated by sympathetic nerve fibers. Strong activation of the renal sympathetic nerves can constrict the renal arterioles and decrease renal blood flow and GFR. Moderate or mild sympathetic stimulation has little influence on renal blood flow and GFR. For example, reflex activation of the sympathetic nervous system resulting from moderate decrease in pressure at the carotid sinus baroreceptors or cardiopulmonary receptors has little influence on renal blood flow or GFR. The renal sympathetic nerve seems to be most important in reducing GFR during severe acute disturbances lasting for a few minutes to a few hours, such as those elicited by the defense reaction, brain ischemia, or severe hemorrhage. In an healthy resting person, sympathetic tone appears to have little influence on renal blood flow. Then, hormonal and autocoid control of renal regulation. Several hormones and autocoids can regulate GFR and renal blood flow. Norepinephrine, and endothelin constrict renal blood vessels and decrease GFR. Hormones that constrict afferent and efferent arterioles, causing reductions in GFR and renal blood flow, include norepinephrine and epinephrine released from the adrenal medulla. In general, blood levels of these hormones parallel the activity of the sympathetic nerve system. Thus, norepinephrine and epinephrine have little influence on renal hemodynamics except under extreme conditions such as severe hemorrhage. Another vasoconstrictor, like endothelin, is a peptide that can be released by damaged vascular endothelial cells of the kidneys as well as by other tissues. The physiological role of this autocoid is not completely understood. However, endothelin may contribute to homeostasis, like minimizing blood loss when a blood vessel is severed, which damage the endothelium and release its powerful vasoconstrictor. Angiotensin II preferentially constricts efferent arterioles in most physiological conditions. A powerful renal vasoconstrictor, angiotensin II can be considered a circulating hormone and a locally produced autocoid because it is formed in the kidneys and in the systemic circulation. Receptors for angiotensin II are present in virtually all blood vessels of the kidneys. However, the preglomerular blood vessels, especially the efferent arteriole, appear to be relatively protected from the angiotensin II mediated constriction in most physiological conditions associated with the activation of the renin-angiotensin system, such as during the low sodium diet or reduced renal perfusion pressure due to renal artery stenosis. This protection is due to the release of vasodilators, especially nitric oxide and prostaglandins, which counteract the vasoconstrictor effects of angiotensin II in these blood vessels. The efferent arterioles, however, are highly sensitive to angiotensin II because angiotensin II preferentially constricts efferent arterioles. In most physiological conditions, increased angiotensin II levels raise glomerular hydrostatic pressure while reducing renal blood flow. It should be kept in mind that an increased angiotensin II formation usually occurs in circumstances associated with decreased arterial pressure or volume depletion, which tends to decrease GFR. In these circumstances, the increased level of angiotensin II by constricting efferent arterioles helps maintain glomerular hydrostatic pressure and GFR. At the same time, through the reduction in renal blood flow caused by efferent arteriolar constriction, contributes to decreased flow through the peritubular capillaries, which in turn increase reabsorption of sodium and water. Then endothelial derived nitric oxide release decrease renal vascular resistance and increase GFR. An autocoid that decreases renal vascular resistance and is released by the vascular endothelium throughout the body is endothelial derived nitric oxide. A basal level of nitric oxide production appears to be important for maintaining vasodilation of the kidneys because it allows the kidneys to excrete normal amounts of sodium and water. Therefore, administration of drugs that inhibit formation of nitric oxide increases renal vascular resistance and decreases GFR and urinary sodium excretion, eventually causing high blood pressure. In some hypertensive patients or in patients with atherosclerosis, damage of the vascular endothelium and impaired nitric oxide production may contribute to increased renal vasoconstriction and elevated blood pressure. Prostaglandins and bradykinin decrease renal vascular resistance and tend to increase GFR. Hormones and autocoids that cause vasodilation and increased renal blood flow and GFR include the prostaglandins and bradykinin. These substances are very important. Although these vasodilators do not appear to be of major importance in regulating renal blood flow or GFR in normal conditions, they may dampen the renal vasoconstrictor effects of the sympathetic nerves or angiotensin II, especially their effects to constrict the afferent arterioles. By opposing vasoconstriction of afferent arterioles, the prostaglandins may help prevent excessive reductions in GFR and renal blood flow. Under stressful conditions such as volume depletion or after surgery, the administration of non-steroidal anti-inflammatory agents such as aspirin that inhibit prostaglandin synthesis may cause significant reductions in GFR. Then tubuloglomerular feedback and autoregulation of GFR. The kidneys have a special feedback mechanism that links changes in sodium chloride concentration at the macula densa with the control of renal arterial resistance and autoregulation of GFR. This feedback helps ensure relatively constant delivery of sodium chloride to the distal tubule and helps prevent spurious fluctuations in renal excretion that would otherwise occur. In many circumstances, this feedback autoregulates renal blood flow and GFR in parallel. However, because this mechanism is specifically directed towards stabilizing sodium chloride delivery to the distal tubule, instances occur when GFR is autoregulated at the expense of changing in renal blood flow. In other instances, this mechanism may actually cause changes in GFR in response to primary changes in renal tubular sodium chloride reabsorption. So when I just try to look at the structure of juxtaglomerular apparatus demonstrating its possible feedback role in control of nephron function, we can say there are glomerular epithelial region, afferent arteriole, juxtaglomerular cells, afferent arteriole, then internal elastic lamina, basement membrane, distal tubule, macula densa region, and smooth muscle fiber. So the tubuloglomerular feedback mechanism has two components that act together to control GFR, an afferent arteriolar feedback mechanism and an efferent arteriolar feedback mechanism. This feedback mechanism depends on special anatomical arrangements of the juxtaglomerular complex. The juxtaglomerular complex consists of macula densa cells in the initial portion of the distal tubule and juxtaglomerular cells in the walls of the afferent and efferent arterioles. The macula densa is a specialized group of epithelial cells in the distal tubule that comes in close contact with the afferent and efferent arteriole. The macula densa cells contain Golgi apparatus, which are intracellular secretory organelles directed toward the arterioles, suggesting that the cells may be secreting a substance toward the arteriole. Then decreased macula densa sodium chloride causes dilation of afferent arterioles and increased renin release. The macula densa cells sense changes in volume delivery to the distal tubule by way of signals that are not completely understood. Experimental studies suggest that decreased GFR shows the flow rate of the loop of Henle, causing increased reabsorption of the percentage of the sodium and chloride ions delivered to the ascending loop of Henle, thereby reducing the concentration of sodium chloride at the macula densa cells. This decreases in sodium chloride concentration initiates a signal from the macula densa that has two effects. It decreases resistance to blood flow in the afferent arteriole, which raises glomerular hydrostatic pressure that helps return GFR toward normal, and it increases renin release from the juxtaglomerular cells of the afferent and efferent arterioles, which are the major storage sites for renin. Renin released from these cells then function in an enzyme to increase the formation of angiotensin 1, which is converted to angiotensin 2. Finally, the angiotensin 2 constricts the efferent arteriole, thereby increasing glomerular hydrostatic pressure and helping to return GFR toward normal. Other factors that increase renin blood flow and GFR: high protein intake and increased blood glucose. Although renal blood flow and GFR are relatively stable under most conditions, there are circumstances in which these variables change significantly. For example, high protein intake is known to increase both renal blood flow and GFR. With a long-term high protein diet, such as one that contains large amount of meat, the increase in GFR and renal blood flow are due partly to the growth of the kidneys. However, GFR and renal blood flow also increases 20 to 30% within 1 to 2 hours after a person eats a high protein meal. A similar mechanism may also explain the marked increase in renal blood flow and GFR that occurs with large increase in blood glucose levels in persons with uncontrolled diabetes mellitus. Because glucose, like some of the amino acids, are also reabsorbed along with the sodium in the proximal tubule, increased glucose delivery to the tubules causes them to reabsorb excess sodium along with glucose. This reabsorption of excess sodium in turn decreases the sodium chloride concentration at the macula densa, activating a tubuloglomerular feedback mediated dilation of the afferent arterioles and subsequent increase in renal blood flow and GFR. This example demonstrates that renal blood flow and GFR per second are not the primary variables controlled by tubuloglomerular feedback mechanism. The main purpose of this feedback is to ensure a constant delivery of sodium chloride to the distal tubule, where final processing of the urine takes place. These disturbances that tend to increase reabsorption of sodium chloride at tubular sites before the macula densa tend to elicit increased renal blood flow and GFR, which helps return distal sodium chloride delivery towards normal, so that normal rates of sodium and water excretion can be maintained.